Okay, I want to walk you through this chapter a little bit. The cell was discovered by Robert Hooke in 1665, and he was looking at cork. And he named it the cell because when he looked at this, it looked like monastery cells all in a row, and so that's why he named it. Also, a glimpse of the microscope, pretty awesome. The cell theory, everything's composed of cells. They're basic units of structure and function, and they only come from pre-existing cells. They don't spontaneously rise like they thought long ago. Size-wise, we can see a lot of things with the unaided eye. This is just looking at that comparison. And down to a frog egg, it's pushing it. Better seen probably with a light microscope, a compound microscope. And you'll see that these overlap all the way down to the electron microscope, that you're going to see viruses and proteins and smaller organelles, so kind of cool stuff. So let's look at some pictures of those. Here's using a light microscope. There's a pine needle, beautiful. Human cheek cells, this sloughs off inside your mouth constantly, and there's the nucleus. There's a white blood cell. Then the transmission electron micrograph, TEM. We're starting to look at things smaller. Here's herpes simplex virans, so those are the viruses. Here's cell organelles, and here are mitochondria. Then the scanning electron micrograph is this new technique, or kind of new, that they actually coat it with gold, and then they get a three-dimensional look like this. So blood cells, looks like some white blood cells are there with red blood cells, and cholera. So pretty darn beautiful stuff. So what determines the size of a cell? Well, surface area to volume ratio. It has to have enough surface area to that volume so that it can absorb everything it needs and get everything out. This big square, this big block, not enough surface area for this much volume. So how does our body kind of make it better? Like the cells in our intestine, we have these tiny finger-like projections that come out of them, just making more surface area. We'll see that coming up again. So size does matter. I have a bad sense of humor. Okay, so we're going to compare prokaryotes and eukaryotes. Bacteria are what you're familiar with, and archaea are these extremophiles that we thought were bacteria, but then we saw their DNA and they're not, but they look just like bacteria to us, but live in these crazy extreme places. Everything else are the eukaryotes. Animals, plants, fungi, protists. So let's talk about those prokaryotes. Prokaryotic means before kernel, and they're talking about before nucleus. So every time you get a question on a test that if they give you an example and it does not have a membrane-bound nucleus, it's going to be one of these guys. It's either going to be a bacteria or an archaea. These guys are small, very successful, all over the place. We think they were the first cells on Earth, actually. Prokaryotic cell is very, very simple structure. We'll spend a whole lot more time looking at the structure of the eukaryotes. So generally what we're familiar with are the bacteria. We identify them by shape. I put this in just simply because it was interesting. Here's anthrax, strep throat, which you're all familiar with, I'm sure, rat bite fever, and Lyme disease are these various shapes of bacteria that we can identify. Okay, what do bacteria have? They have a cell membrane. Who else has it? Eukaryotic cells. Everything has a cell membrane. At the end of this, I'll be talking about cell membranes in detail and how they work. This is a cell membrane right here. So remember that everything's got it. It's a phospholipid bilayer with embedded proteins. We'll talk more about that soon. So the cell wall. The cell wall of bacteria maintains this shape. And an interesting thing here, it's made up of peptidoglycan and in different quantities if it's a different kind of bacteria, gram-positive, gram-negative. You might have heard that before. So penicillin doesn't let this work as well, and so these guys die. Doesn't impact gram-negative. And so that's why they'll check to see what you have if you have a culture done, if you have an infection. Cytoplasm. It's a semi-fluid inside here. It's got inorganic, organic molecules. It's a lovely mix of rough resources to use. And they have a nucleoid region. It's called nucleoid, sorry, I'm stuttering. It's not a true nucleus. It's a location where the DNA strand is located, but it is not a membrane-bound nucleus. And so that's what makes these different. However, they have ribosomes. They're smaller than the ones in the eukaryotes, but eukaryotes